Hi everyone, my name is Cha Cha. Welcome back to my channel. Once again, I am a student who majored in psychology and molecular biology. So there may be some mistakes in my channel, but if I did make mistakes, please let me know because I'm also studying for the MCAT and I really want to know if I screwed something up because it's important for me. Today I'm going to be going over the first chapter of in the Kaplan textbooks for biology, which is the cell. Without further ado, let me scroll up and get to it. So cell theory, there's like four core pillars of life that I like to think of. There's three that were kind of older, and then the fourth one is kind of added on later when scientists kind of figured everything out. The first one is that all living things are made of cells. Cells are the basic unit of life, which kind of go hand in hand. So it's not atoms, not molecules. Since if you think about like sodium isn't alive or water isn't alive, it has to be a specific cell itself, which is made out of atoms and molecules um, they're more on a macro scale than those are. Cells arise only from pre-existing cells, which is kind of a paradox since if you think about it, how did the first cell arise to begin with? And yeah. And then the last one is that cells have instructions in the form of DNA. Scientists didn't originally know what we were made out of originally. Uh, there's a lot of interesting things that they used to think about what we were actually made of, not DNA. That is definitely not one of them. Um, but so I sort of think about it as like a family recipe that's being passed down. So sometimes you can modify the recipe a little bit and taste great. Like maybe your grandma had a recipe and she didn't add enough cheese, but you wanted to add more cheese to your lasagna. So that's great. It's tasty. You slightly modified it, but it's essentially the same dish. Otherwise, it could taste like cardboard. Like, let's say that you replaced sugar in your cookies for salt. That doesn't taste very good at all. Fundamentally, it is still the same cookie, but it is a deleterious, I should say, mutation. Or, um, then let's kind of go into more of eukaryotes versus prokaryotes. So those are like the two main classifications that the MCAT will be focused on. So eukaryotes, they have a true nucleus, so it's a separation of transcription and translation, whereas prokaryotes, there are no organelles. So the only double membrane is between the inside of the cell and the outside of the cell. So that means that transcription and translation can occur simultaneously, which is pretty interesting that that can happen. There's not really as much regulation going on. Eukaryotes are typically larger, so they're usually cellular. They can also be unicellular too, but that's more rare, whereas prokaryotes are typically single-celled only. Eukaryotes have, as I was saying, they have organelles, so they're a lot more rigid, more complex. So it's important for molecules to be further apart, and like to have clear regulations and instructions. Whereas prokaryotes don't have organelles. It still allows for quick diffusion, given their size, because they're so small, molecules can just get from one place to the other really quickly. So everything's just like boom, boom, boom. Um, Whereas eukaryotes is more organized, they're kind of more anal in a way. So as I was saying, eukaryotes, they have separation, they have organelles, they have other places where there's that phospholipid bilayer, which is typically how you separate hydrophobic and hydrophilic. Aspects are just like itself. It's kind of like like in movies where they have like laser fields where you can't like get through to the other side. That's kind of how I like to think about organelles and a phospholipid bilayer. The mitochondria has inner and outer membrane, and the ETC occurs, or electron transport chain, which is like where you get most of your ATP due to the ATP pump. That occurs in the mitochondria. Whereas in prokaryotes, because there's no organelles, it actually occurs in the cell membrane itself. There is no mitochondria in prokaryotes. Eukaryotes, their DNA is double-stranded linear. Goes from like 5 prime, 3 prime, and whereas for prokaryotes, it's circular, so it's continuous transcription, and it's less vulnerable to degradation. So Eukaryotes, they have telomeres, and telomeres help aid in DNA replication, not to confused with transcription, which is completely different. Transcription is when you're trying to make a protein. DNA replication is the S phase or synthesis phase, which happens during interphase of mitosis. That's just like where the DNA itself is being replicated so that cell can divide and create another version of itself during mitosis. Mitosis is when the cell just creates two copies of itself. So it's like boom, boom, I guess. Um, whereas, yeah. Uh, and Telomeres are really important because they kind of have nonsense. They're not ever going to be kind of like encoded by anything, I guess. They need to be somewhere for the RNA primers to start, otherwise you'll lose some of that precious DNA that will can actually code for genes and is important just in intergenic regions itself too, since there's a lot of important stuff in intergenic regions like enhancers and it can bind to yeah, it's just somewhere to bind, but they shorten during each time the cell replicates. Typically there's around 30 or so I guess I should say times that the cell can actually divide before the telomeres have degraded so much that it starts to interfere. So that'll signal an apoptosis signal. Yeah. Um, yeah, whereas over here, this is a lot more specific. Prokaryotes, they can only go via asexual reproduction. So like conjugation, binary fission, transduction, etc. Whereas for eukaryotes, can only be sexual, which is typically in the gonads, or only in the gonads, I should say, meiosis, which is where you take it from a parent to parents, and asexual, which is mitosis, which is I was sort of saying like those are your somatic cells. So that's when you're just having like one cell is completely identical to what its daughter cell is, whereas meiosis, that's not the case. Okay. Um, So, as I was saying, there are organelles in eukaryotes, whereas there are no organelles in prokaryotes. So, it's kind of important to talk about the main ones that are important for them. Cat. Also, over here, I just kind of have the central dogma. I don't know why it's called the dogma, since I just think of like dogs. It doesn't really have too much. But DNA occurs in the nucleus. And then it has a signal like once it has becomes mRNA. So mRNA kind of has a transport signal which tells it to exit the nucleus to go into the cytoplasm after it gets transformed into RNA. And it has like that 5' cap and poly A tail on it. The poly A tail is actually that signal once it gets transformed to mRNA, then it goes out and then it can go into the cytoplasm. It can get translated and it becomes a protein. So also kind of how I like to think about like transcription and translation. Like when you transcribe something, it's the same language. You're just like kind of like maybe going from like um I don't know, like writing like a manuscript of a book on paper to typing it. Whereas once that book is like fully typed, maybe it becomes a best seller, so you want to translate it into different languages. And that's kind of how I like to think about DNA, RNA, proteins. Anyways, that's kind of getting ahead of myself. The nucleus, that's pretty important. Um, That's where transcription occurs. So it's kind of like that DNA to RNA. And there's also different types of RNA. So there's rRNA, which is the ribosomes. There's tRNA, which is really important for having the amino acids that bind to it, which is part of that protein or is part of the protein. And then there's mRNA, which is the messenger RNA, which is sort of like the manuscript for, or sorry, DNA is the manuscript for RNA. It just does its bidding. It's just like, um, pretty similar. It's just the instructions for the DNA. It's like a kind of a game of hot potato or like telephone. So DNA, RNA, protein. Um, yeah, it has a double membrane, so that means it's extra secure. Only like once I have like double membranes, it just basically means that it doesn't want to get out. Same with the mitochondria. Those are some of the only organelles that have double membranes. Um, it is kind of like a highly monitored prison. It's hard, and a lot of stuff needs to happen in order for stuff to break in and out. So you kind of need a badge. So like think about it, you're like going up to like Mr. Prisoner, and then you like badge in. I don't know. It's like, oh, yay, now I can go in. Woohoo. But it's very high tech. Need to badge in, which is kind of that nuclear localization signal to get through the nuclear pores, which in this case are the prison gates. Um, the nucleolus is a subsection of the nucleus where the rRNA is made, so the ribosomal RNA. The mitochondria, I'm sure, as everyone has heard since they've been in sixth grade biology, maybe even younger, is that it is the powerhouse of the cell. Woohoo! Is where the majority of that ATP is made due to the ATP pump. It also has that double membrane, so it's really important. It kind of has that like prison system going on here. Its outer layer protects the mitochondria from the cytoplasm, while the inner membrane forms cristae, and cristae, like which I kind of crudely showed over here, are like these folds. So it's really important for increasing that like just like surface area. It's because like of the electron transport chain. Um and hydrogen pumping kind of between the inner membrane space. So the inner membrane space would be like uh this is the outer, this is the inner membrane space. This is the inner membrane, and this is the matrix, mitochondrial matrix. Um, yeah. Mitochondria are also interesting because there's like different theories, I should say, about the mitochondria. Some scientists believe that like millions of millions of years ago, there's probably some eukaryotic cell that actually ate a prokaryotic cell. And I was like, this actually tastes good, and it's doing something cool. It's doing an extra job for me that I don't have to do anymore. Um, and like this whole electron transport chain like just kind of happening inside of it. Wow, I like save energy and I'm creating like a lot of energy. I'm doing all this stuff. So it's unique because it has its own genes and transcription elements. So that's why scientists believe that some eukaryote like um phagocytosis are like kind of enveloped and ate this prokaryotic cell millions of years ago, and it just kind of stuck because it's efficient. Which is interesting. No other organelle has its own unique set of genes and transcription elements or double membrane. Double membrane rise up. Kind of interesting, maybe, just because it wants to protect itself. Then there's lysosomes. Lysosomes have a super acidic pH due to hydrogen pumps. So as kind of it's very similar how it works to like the ATP pump where it has like the hydrogen pumps that put hydrogen instead of like into the matrix from the inner membrane space, it does it into the lysosome. And because it's such an acidic pH, remember proteins and just like everything kind of poorly reacts at lower acidic pH. It leads to degradation and for elements to get destroyed, where the cell can actually reuse those elements. Since we're all about efficiency here, we don't want to use any more energy than we have to. The endosome is kind of like the packaging center for the cell. It's the sends components where they need to go. It's kind of like a mailroom, sort of, and it does this often via vesicles. Here I kind of crudely drew like a vesicle. Usually it kind of has um, like a hydrophilic shell and then a hydrophobic center. Typically, since if you're in a vesicle, you usually are hydrophobic. Yeah. And it has like these little compartments for molecules to like kind of sit. And it's like an airplane. That's kind of how I like to think about it. Goes from one place to the other. The rough ER, it's studded with ribosomes, hence the name since it's rough, quote unquote. It's important for folding proteins, so it has heat shock protein 70, like that's like a common one that you'll hear of, or just like heat shock proteins in general. It's really important for the unfolded protein response. So if your cell is under a lot of stress, it can start folding upon itself with these extra chaperone proteins, which can hopefully help refold these proteins. And if it gets too overwhelmed, it could actually lead to apoptosis. Um, yeah, it's really important since like it'll bring proteins that are hydrophobic, like or membrane or secreted proteins, they will fold here. Um, Yeah, it's attached to the nucleus, so it also has a double membrane, and sugar modifications can also happen in the rough ER. The smooth ER is important for making lipids and detoxifying poisons. So, it's like fatty acid synthase occurs here. There's no ribosomes on it, so it's as smooth as a baby's bottom, as we like to say. Yeah. Um, then we have the Golgi. And the Golgi is also another interesting one. There's a lot of theories about how molecules actually move through the Golgi, but I'm not really gonna get too into that. Here we're going back kind of into our vesicles. So between the ER, the rough ER and the cis Golgi, so cis Golgi is the newest like molecules are like where they first enter the Golgi, and the trans are when it's like the final modification, so it's furthest from the Golgi. So COP1, that's just like the name of the vesicle. That goes from the cis Golgi to the ER, whereas COP2 goes from the ER to cis. You think it'd be the opposite naming, just like a lot of stuff that happens in biology. COP1 was found before COP2, the specific type of vesicle, and they're just named that because of the proteins that are on the vesicle's surface. Um, yeah, it is kind of young. It's like many modifications as it moves through the Golgi, whereas trans is for list, and that is where they are exocytosed via clathrin, which is the other type of vesicle that you'll most likely hear on the MCAT, and clathrin is just important since it goes to the cell surface. And it is into the extracellular space. Then there's the peroxisome, which is aptly named. They got really creative with the name here because of hydrogen peroxide. And it's also important for lipids breakdown and synthesis. Okay, now we're going to go on to the cytoskeleton structure. So just kind of like what makes a cell kind of look the way it does and everything else inside of the cell. So you have microfilaments, which are really in like actin. That's like the number one thing you'll hear. So like that's important for like neurons and stuff like that. That's like a big thing that makes up dendrites. It's also important for muscles, so contraction, relaxation, and cytokinesis during replication. All right, I'm just gonna water. Then you have microtubulin. Um, and tubulin has like the GTP, GDP cascade. It's just sort of like a ever-moving, like kind of conveyor belt, I guess. Like you have certain parts and it just kind of like builds upon each other. It's kind of like a highway, conveyor belt, whatever you want to call it, um, of the cell. So it goes typically from like the center of the cell, towards the exterior. And it's just like a whole bunch of like highway system. It has motor proteins, which has um yeah, which are like these like funny little things that can like walk around to like kind of think about it like trucks that deliver molecules from one end of the cell to the other. Um, they also make up cilia, which is just like help move stuff around the organ tracks. And flagella, they're kind of like propeller like structures, which uses torque to move. Like I have like the little propeller hat here. That's kind of like how I think about flagella, and it uses torque to move stuff around. And it also has centrioles, which is really important for pulling apart stuff of the cell and the chromosomes during replication. Then you have intermediate filaments. They're not really as important. They are important for cell-to-cell adhesion, structure, and they're kind of like a jack of all trades. Then we have tissues. Um, you have the epithelial, which cover the body and the cavities. They're involved in absorption and they're attached to the basement membrane. So they are polarized. So you have the outside and then you have the lumen. Lumen just is kind of like the opening inside of the cell, or not inside, so like just like inside of like tracks and just different stuff. So it just like faces like inside, not like the circulatory system typically. Um, then you have like there's different types of epithelial, just because epithelial is like such a broad category. But here I'm gonna go over kind of like the shape, and shape is like really important, and also whether or not they're stacked on top of each other. So like whether they're a single layer, they're stratified. Stratified just like essentially means that they're like the stratosphere, kind of think about it like that, where there's different layers. And then pseudo-stratified, pseudo meaning kind of like fake almost, or like kind of like a magic trick. That's why I like to think about it. So it looks like it's actually layered, but it's not. Then there's cuboidal, and cuboidal is they look like cubes. Columnar, they're kind of long, they're more vertical, whereas squamous are kind of squished together, and they are more horizontal. So, helps with the parenchyma, which is a functional part of organs, like the mushy parts, like the stuff that like you squeeze or whatever. Um, and then there's connective tissue, and connective tissue is what helps support structure, like bones, cartilage, like tendons. So tendons is when you go from like muscle to tissue. Then we should go to prokaryotes since we talked enough about eukaryotes. Archaea. Here are the two different types of domains that you need to know for them, Cat. There's archaea, which live in extreme conditions. They have alternative sources of energy, but they're kind of similar in structure-ish to eukaryotes. Then there's bacteria, which have cell membrane, cytoplasm, flagella, like their ribosome is small relative to eukaryotes, which makes sense. Just think about like prokaryotes are smaller in nature. They're unicellular versus eukaryotes, which are typically multicellular, and they can be parasites or they can be mutualistic symbiotes. So over here I kind of have uh like parasitic relationship where one or his person is like giving more than the other, whereas this is more mutualistic. They both kind of benefit. So like an example from like a parasite would be like something that's like necrotizing or something that's like kind of like eating the host. Like if you think about like a dead tree, that's like in the forest, or if you think about mutualistic symbiotes, you can think about nitrogen fixers in plants. So like nitrogen fixers get all of like the good stuff from plants, like a lot of like enriching nutrients, whereas nitrogen fixer like the bacteria in plants, they get to break down nitrogen since nitrogen has like N2 looks like this. So very hard to break this triple bond. Lots of energy required. And they are a specific type, like in their the specific bacteria can break that triple bond. Um, so then there's different types. So they're not like domains or anything. They're just important to know for bacteria. So there's aerobes. Aerobe, like think about like aerobics, like you use a lot of like oxygen like when you work out, I guess. So it require like oxygen metabolism. And then there's anaerobes which don't use oxygen for their metabolism. So in humans, this would be like glycolysis, anything in your cytoplasm doesn't require oxygen, so glycolysis doesn't. Like lactate, like lactic acid, like that type of stuff doesn't either. Um, and then you have obligate aerobes, which require, so like they're obligated to use oxygen. That's kind of what I like to think about it. Then there's obligate anaerobe, so it dies in the presence of oxygen. So like they require they're obligated to like not use oxygen. Then you have facultative anaerobes, so they are switching between oxygen and metabolic pathways that don't require oxygen. Then there's aerotolerant anaerobes. They can survive oxygen but prefers like no or minimal oxygen. That's the name aerotolerant, like they're tolerant. Then we'll get into kind of like the structures of prokaryotes. So there's a cell wall, which is an extra layer of protection. It's sort of like the skin or ligamentary system of prokaryotes, and it's only like um until then there's the envelope, which is the cell wall and the membrane. So like the cell wall is kind of like extra like support, I guess. Um I guess it's more like the bricks, and then the cell membrane is more like the soft interior, or like the drywall. Gram positive, they are easily accessible. They're different types of bacteria, I should say. It's easily accessible, so open to pathogens. So like if you were to have like a dye. Like let's say you have like they have like the purple dye test, like kind of to determine between gram positive and gram negative. So if you have a purple dye and you were to input it into like gram positive, let's say that the gram positive originally actually I think of the color. Yeah. So like you have gram positive in here. Let's say this is originally um like red inside. And then you add a yellow dye. So the yellow dye can penetrate inside, so it becomes, as a result, more orange. Ah, as a result, the inside. Whereas gram negative, it's harder to penetrate. So it's negative, like mission failed, sort of. It's gonna stay that red color. It's not going to change. Um, like the dye can't penetrate inside. So the end result is that it's still red inside. Um, Flagella, we kind of already talked about that. That allows the cell to easily move around. And then plasmids are external sources of DNA, so they are not originally part of the genome. They can become integrated. So a lot of the times you'll have plasmids are usually like really important tools. And there's a lot of like selection purposes too. Like for bacteria, you could have an specific antibiotic included in your plasmid and also have a gene of interest or something that you want the bacteria to have or manipulate. And if the plasmid is not absorbed by that bacteria, if you put it on a plate with that specific antibiotic, then those without the antibiotic resistance will die. So, you only have the bacteria that have the plasmid incorporated into it actually grow. That is a very common thing, and I have done that before. Reproduction. Woohoo. Okay. Massive, massive. We're moving on. So this is for prokaryotes. Binary fission. It's more rapid mitosis, so it's quick doubling time. So, if you think about like E. coli, they double every like 20 minutes, which is really fast. So you have the lag phase, which is like originally, they're like starting to like kind of get used to their conditions. They're almost like starting to double. The log phase is when they double. The stationary phase is when the resources available in the doubling time like they start to level off. And then the death phase, like kind of think about like the Thanos phase. It's like when the maximum resources have been reached, they can no longer grow. So they start dying. And this is called a sigmoidal graph, at least this part until they start dying. Um yeah, kind of because it kind of looks like an S. Yeah. And so yeah, that's just like when it's just like rapid mitosis sort of. Um like fission, think about like fission like the star is like breaking apart, like doubling. Um, Genetic recombination is when plasmids are integrated into the genome, kind of what I was talking about up there, about like bacteria and antibiotics. Testing, then there's transformation, which is when foreign genetic material enter the genome. Conjugation is when you actually have an exchange of information from two separate bacteria, except for this is technically not considered sexual reproduction though. So over here we have two bacteria, look at them with their little flagellas and stuff. They have a sex pili, which can transfer like genetic information, so DNA, from one to the other. So this little blue cell will have now that DNA um through that specific through the sex pili and just sort of like a bridge to exchange information. Transduction is when new information is introduced to bacteria via viruses by vectors. It's accidentally done by bacteriophages since like viruses they need a host to reproduce. They're pretty parasitic in nature. They aren't considered alive because they use typically use RNA. Not all, but most use RNA um to replicate, and a lot of times they'll hop from bacteria to bacteria. So eventually, like sometimes they'll actually bring some of the genetic information from one bacteria to the other. There's also transposons, which transposons actually also occur in humans too. And transposons is when it's just like random jumping around of like the genome. So like one part of the genome goes to a different part of the genome, and this can actually disrupt genes. And it's completely random. It's up to chance. Viruses, as I said, they need to replicate via host cell. So, if you think about like an alien where like the alien bursts out of his chest, they're kind of the same way, like they need a host. And a lot of times it actually kills them, just like an alien. That's called the lytic phase. So lytic kind of like lysis, when you think lyse, it like break apart. The lysogenic cycle, the virus can integrate into the genome, like kind of like the TV show, like a like the parasite. Um, it is made of DNA or RNA, and it's not considered alive, which is a indication between all of these. The viruses are the only type that you'll see on the MCAT, um, that is not live. Positive sense viruses are single strand positive sense virus. It's viral RNA, which can be made directly into proteins, so it doesn't need any other like cellular mechanisms. It's just like mRNA, sort of where it could just be directly made into proteins. Negative sense viruses are viral RNA, which are in the opposite orientation. So instead of like being from like 5 prime to 3 prime. They're like 3 prime to 5 prime. And they act as the template strand. So it needs to bring in its own RNA dependent RNA polymerase in order to become like in the positive sense direction. There's also retroviruses. And retroviruses convert RNA into DNA, and it also transmits reverse transcriptase to have RNA into DNA. It is kind of like a permanent fixture of the genome. So a common example of this is HIV, where it'll like the virus will actually integrate itself completely into the host genome, and this is carried on in other cells. So like when it that cell undergoes mitosis or like DNA replication, during the S phase, it doesn't get caught by any of the specific mechanisms in place to catch any like mutations or anything, because the host recognizes it as part of its genome. Some various things to know is that there's prions, which trigger misfolding. Prions are really scary to think about because there's no known treatment. Example of this is mad cow disease. It's highly contagious, so even if you touch something that has prions on it, chances are you're already infected. It goes from alpha helixes on. Alpha helixes are typically found in membranes, so they're more hydrophobic to beta sheets, which are more hydrophilic, so it manipulates solubility. There's also viroids, which are single-stranded RNA, which infects plants. That's kind of all I had on this. I know this was all quite a lot. The cell is lots. There's a lot going on here. Yeah, thanks again for watching. I hope that you guys enjoyed. Once again, my name is Cha Cha, and let me know in the comments how you liked my video.